Hello everybody. Right now I am into the home page of the course Science 230 Introduction to Bioinformatics, and my name is Dr. Ramneet Kaur. And first of all, let me give you my own little background and my introduction. So I am PhD in biotechnology. I did my postdoctoral research fellowship in the field of cancer biology from Harvard Medical School, and I have worked in the Emory University for 2 years in the Winship Cancer Institute. Currently, I'm teaching at Mercer University. I also teach at Georgia Gwinnett College and at Georgia State University. At Georgia State University, I'm also conducting research in the field of cancer biology. So I will be your instructor for this course, Science 230 Introduction to Bioinformatics. So this is the home page of the course. So if you can see that on the left side, I have created some folders. So there is a course overview, course content, lab Dropbox, assignment Dropbox, course information, then course announcements, grades, calendar, student support. So all these are your folders which are you going to use. So I'm going to go over all these folders and tell you what is inside every folder, and then I'm going to discuss your syllabus with you. So course overview, if you can see the course overview. So basically in the course overview, it basically tells you, it's an overview of the course. It tells you what is in your course information, what is in your course content, what is in your course announcements, all these things. There's grades, email, if you want to email somebody. Then if you see this course content, you can see here in the course content, I have created 8 folders. So week 1, 2, going up to week number 8. So every week you will be going into these folders, and you will find your stuff, whatever stuff you have to do for the week will be in the folder. So, and also you can see outside the folder, kind of a statement will be there, what we are going to do this week. So this week we will talk about molecular biology and evolution. So in the first few weeks, I'm going to make a background about biology, because it's very important to know about biology first. Only then we can apply those computer tools. We can learn about those computer softwares. So this week, you can see I'm in the week 1 folder, and you can see all these things here, and I may be adding few more things here. So this is your lecture number 1. This is my PowerPoint. This is my study guide. So this is my PowerPoint for the lecture number 1. This is the study guide. And right now, I have also created a video lecture for you guys. And that video lecture is right now getting uploaded on YouTube. As soon as it's uploaded, I'm going to put the links here, and you can watch those videos. And also with so it will be every week. I will give you PowerPoint, I will give you study guide, I will give you my video lecture, and also I will post some additional videos. You can see I have posted here 5 videos. So this is my additional material, because by watching these videos, you will have a great understanding of the subject. So I want all of you to watch these videos, because that's part of the lecture, to understand the subject. And coming here, you can see here there is a virtual lab number 1. So we will be having basically 2 kinds of labs in this course. There will be wet labs and the computer labs. And in the wet labs, there will be basically we have 7 labs in this course. 3 labs are computer labs, 4 labs are wet labs, and out of those 4 labs, wet labs, 2 labs are going to be virtual labs, and 2 labs are where you're going to get hands-on experience. And to do those labs, you need to buy a kit from Carolina, and the information is in the syllabus. So please buy those kits as soon as possible, because to get the hands-on experience, those 2 labs will be using the kit from Carolina, and we will be needing the Carolina kit in the second week. So please buy it as soon as possible. You will be needing it next week. So this week the lab is a virtual lab, and it's about extraction of DNA. So you are going to watch that lab, and you will be actually working in the lab, and you will be in a virtual lab, and you will be extracting the DNA. After you are done extracting the DNA, here is a worksheet for you. You have to answer some questions. There are 4 questions in this worksheet. You will be answering those questions in the worksheet, and you will be submitting that worksheet here. I have created a Dropbox for the labs. So this is a lab Dropbox. So you will be submitting your worksheet here. And then after every lecture you have watched, there will be every week there will be one quiz and there will be one assignment. So the quiz will be on Blackboard. You will be going in the quiz, you'll be entering into the quiz, answering the questions, and you're done. Another thing is an assignment. So every week there will be a writing assignment. And what you need to do in a writing assignment, in the writing assignment, basically you have to apply whatever you have learned in the lecture. You have to apply that into your daily life. So basically, which means that if you have learned about DNA, RNA, or a protein, you have to write how that information is applicable in your daily life. And that assignment, you have to write 1 to 2 pages, and you will be submitting that assignment. So it will be every week. That assignment you are going to submit here in the Dropbox, which is the assignment Dropbox. So in those assignments, you're going to link theory to practice. So basically every week you will be working on assignment, quiz, and the lab, and you will get 1 week time. So all the assignments will open on Friday in the evening, 5 p.m. and all these assignments are going to be closing on Thursday night, 11:59 p.m. And the reason is I have to post the attendance in on the Friday morning, so I need to know, and the attendance will be on the basis of the submission of the assignments. And also this week you have to do another training, which is the orientation for online learning. So this is, I have posted here the instructions how to get enrolled into this training. So please, you need to do this training, and you need to this is a kind of a survey, feedback survey. You have to do that, and for that also you have a time period. You have to be doing that by next Thursday night. So the quiz will be here. Okay, so this is basically your course contents. So there are 8 folders. Every week's stuff will be in the respective folder. So this is lab Dropbox, where you are going to submit your labs worksheets. Here is the assignment Dropbox, where you are going to submit your assignments. And then we have course information. So if you go inside the course information, you will find here a syllabus. And now I'm going to discuss with you your syllabus. Okay, so it is getting downloaded. Just 1 second. So it's here. Okay. So the syllabus is opening right now. And in the syllabus, I'm going to discuss how we are going to function in these 8 weeks and what we have to accomplish. Few more seconds. It will be in few more seconds. So in the syllabus, I'm going to discuss with you which book we are going to use, and the kits you need to buy from Carolina. So for this course, basically I have created my own material, and that will be the most important for you guys. Okay, so this is the syllabus. So you can see here, this is my name, and here is my phone number and my email. So please, if you have any problem, please don't hesitate to contact me, and I will be very happy to help you. So please, anytime you have any doubt, any concerns, please let me know about it. So okay, it's still working. Okay, so this is the course description here. So in this course, basically hold on 1 second. Oh my god, what happened? So in this course, basically what we are going to do, we are going to learn first of all about biology part, and then we will be working with the various softwares. Okay, so in this course, you will be doing various kinds of analysis. You'll be doing protein analysis. You'll be doing DNA analysis. In this, you will be doing various protein modeling. We'll be using various protein models. We'll be finding where the protein is present in the cell. We'll be finding first protein is in the membrane. What part of the protein is in the membrane? We'll be finding different domains in the proteins. So basically, the overall description of this course is student will be introduced to contemporary concepts in molecular biology and evolution, employ the computational algorithms used in biological data based searches, protein and data sequence analysis, prediction of protein functions, specific methods, tools, technologies examined in the course will include but are not limited to detection of homology with BLAST. So we'll be using all these softwares, all these tools are present on NCBI site. So we'll be using from there. And BLAST is a tool where we can find homology of the protein or a gene with various other proteins and genes, whatever they are in the database. So we can basically compare some proteins. We can compare two proteins. We can compare multiple proteins. We can find domains in the proteins. We can find where the protein is localized. So you can do all these things if you have a good knowledge and information and some background about biology. So that's why I will build your I will give you some nice background of biology, and then we will move to the computer part, software part. So basically this is the book. So this book is a recommended book, so you can use this book if you want to refer, but I have created my own material, and my my PowerPoints are going to be the main thing in this course. So it is optional if you want to buy the book, because my PowerPoints are the main thing. So it's a recommended book, it's optional. Now second thing is lab kits by Carolina. Now this kit you need to buy. I think it is worth $82 or $86. This you have to buy because there are 2 labs you will be doing using the material from this kit, and this is a website. You can go here and you can order this kit, and I will request you to please order this kit as soon as possible because you need this kit in the second week. We will be using Blackboard for all our work, and I hope you guys know how to use a Blackboard. And if you guys have any problem, you can contact, these numbers are given to you. Required material you will be needing for this course. You need internet, you need the computer, and this is an online class, so there are some policies. So if you have any problem with any grades, you can always talk to me. And please, whatever the class schedule is there, that's tentative, it can change. Please submit everything on Blackboard. Do not send me anything by email. Everything has to be submitted on Blackboard, and everything has to be submitted in 1 week's time. All the assignments, all the quizzes, all the Dropbox, they will be closing on Thursday night 11:59 p.m. and new assignments will be opening on Friday 5 p.m. So please be in time. If you have some kind of emergency, and the situation is written here, you can see that in that case only some quizzes can be made up if you provide a official documentation. No labs can be made up in any situation, and no late assignments will be accepted. So you have to be in time, you have to do everything in time. And also your attendance is going to be on the basis of your submission of the assignment. So that is why I will be closing all these drop boxes on Thursday night 11:59 p.m. because I have to report the attendance on Friday morning, and that I will be doing on the basis of the submission of the assignments. So if you don't log in and don't do the assignments there, you will be marked absent. So final grades are lowered by 1 letter grade if the student does not log in 25% of the scheduled online time, which means if you are absent from the course for 2 sessions, your letter grade will be lowered by 1. Suppose you're getting A, because you have missed 2 sessions, you'll get B grade. So please, attendance is also very important. Okay, we have discussed this. Okay, whatever stuff you are posting there, don't plagiarize. We have ways to check it. and plagiarism is considered academic dishonesty, and it can be reported and you will be failing this course. So here is some policies. Online community of respect, you can go over these. Online academic integrity. Disability, if you have any kind of disability and you need any help from me, please let me know. And here is the disability office at Mercer. You can always check with these people also. Grading policy, you know what grading policy is. If you are between 90 to 100, you have a A grade. If you are between 85.1 to 89.9, you have B plus. If you're between 80 to 85, you are B. If you are between 75.1 to 79.9, you are C plus. If you have between 70 to 75, you have C grade. If you are between 60 to 69.9, you have D grade. And if you have less than 60, you fail this course. And here is the grading formula. So basically there are 5 components. The lab work, all the worksheets. So basically in this course we have 7 labs, 3 computer labs and 4 wet labs. Out of those 4, 2 are virtual labs and 2 are which are going to give you hands-on experience, and for that you need to buy the Carolina kit. So we have, from every lecture we will be having quizzes. Quizzes make 15% of your total grade. There will be 6 quizzes. Then writing assignments, in which you are going to link theory to practice, and these assignments will be in the form of blogs, journals. Every week it will be a little different. So but basic concept is to link theory to practice. So these assignments, there will be 6 assignments and they are worth 5%. Also at the end of the course you will have final exam, which is worth 25%, which will have all the questions the way you will be getting during the session. In the quizzes you will get similar questions in your final exam. And the most important thing is the final project, and this final project is worth 30%. So what is this final project? Around 5th week I will give every student a name of a gene, and whatever you are going to learn in the computer labs, you have to use all the computer softwares, all the computer tools, whatever you have learned in those labs. You have to apply all those tools on that gene, and you have to make your own project. And last week you have to submit your project in the form of PowerPoint. And I will also provide you with the instructions how to work on this project. So around 5th week I will give you the name of the different genes, and I will be providing you the instructions how to work on this project. So for each lab, both wet as well as computer lab, worksheet will be provided, and the completed worksheet will be graded. You will get 1 week time to complete the worksheet and submit. So 1 week time, everything closing on Thursday night 11:59, and also you have to submit your labs in the lab Dropbox. Okay, so we will have a quiz. We will have a quiz from every lecture, and you will get 1 week time to finish the quiz. Every week you will be required to write 1 to 2 pages, around 300 words, the application of theory you learned in every lecture in your daily life in the form of written assignments, journals and blogs, and again you will have 1 week time to submit. The time for submission of all assignments will end every Thursday night 11:59 p.m. We will also have some online interactive activities in the form of journal club, where we will discuss the research paper. So all these interactive activities we will have, especially the journal club, we are going to discuss the research paper. It's not going to be graded, but it's going to help you for your final project and your final exam. There will be a final exam at the end of the course. In the last week, students will also submit their final project in the form of PowerPoint presentation. The project will be assigned by Dr. Kaur to each student. The project will basically involve the analysis of some gene using bioinformatics tools, which students will learn during the course. So this is the grading formula, and this is our schedule. So this is a schedule for 8 weeks. In the week number 1, so remember that every week you will have a quiz, you will have assignment. So we have 6 quizzes, 6 assignments. The quiz will be there, and you will enter into the quiz, do that and submit it. Assignments will be submitted in the Dropbox, assignment Dropbox, and there will be around 7 labs. So all the worksheets from the labs are going to be submitted in the lab Dropbox. Okay, let's see. So this is a pattern we are going to follow for every week. So in the first week, we will be learning about molecular biology, basically, which means we're going to talk about DNA, RNA, and proteins, and the central dogma. And we will also talk about evolution. The lab, first week is a virtual lab, and it is about extraction of DNA. Second week, we will be learning about the technique called DNA sequencing. So how DNA sequencing is done, and the lab for the second week is on hands-on experience. For this lab, you need to use the Carolina kit, and the second lab is about DNA, RNA, and the central dogma. Third week, we will be learning about DNA microarrays, and the virtual lab is about the DNA microarrays. Moving towards fourth week, we will be learning about what is blast analysis, what is pairwise and multiple alignment techniques. We'll be learning about these, and the computer lab number 1. So we will start here, computer lab number 1. It will be on blast analysis. So we'll see what blast analysis is, what is pairwise, multiple alignments. We'll learn all these things, and in the lab we will actually practice the blast analysis. Fifth and sixth week, we are going to talk all about proteomics. We are all going to talk about proteins in the lectures. And in the lab number 5, which is the computer lab number 2, we will do pairwise and the multiple sequence alignments. And in the lab number 6, which is computer lab number 3, we will learn about the protein analysis tools. So for all these computer labs, I will provide you the worksheets in which the exercises will be there, and you will work on these, and I will provide you with the video lectures for these, and they will be really helpful to you to solve all these problems. Or maybe we might use the Blackboard Collaborate for doing the I will see, I'm not sure how we will do, but maybe we will be using this Blackboard Collaborate, and I will let you know 1 or 2 weeks before, so that you can also arrange, because for that we need the webcam, we need the mic, because that is actually a virtual classroom where we can chat, we can see each other. So maybe for computer labs we may use Blackboard Collaborate, or even we can even do the video lectures. Both ways are effective, so I will let you know how we are going to do. So in the third lab, in the protein analysis, we will be doing a lot of things in this lab. We will be making models of proteins. We will be finding where the protein is present in the cell. We will be finding various domains in the proteins. We will be finding if some protein is in the membrane, what part of the protein is in the membrane, basically, which means we will be finding the transmembrane regions. Seventh week is a discussion of a research paper. So we will be discussing a research paper, and this activity is not going to be graded. And the lab here, you will be using the Carolina kit, and here there's experiment, introduction to biotechnology methods. You will be working on this on the 7th week. Last week, week number 8, is your final project time submission, and also you will be having the final exam. So here is one very important note from me. For the lectures, I will be having information from various sources. All my PowerPoints along with my video lectures will be posted on Blackboard. I might also provide some extra material for your reading. My PowerPoint presentation will be the most important for the course. So book is optional. You want to buy the book, you can buy, but I have created my own PowerPoints, and these PowerPoints are from various sources, and also I will provide you with some extra reading material. And also I will be, every week I will be also providing you with lecture with the videos, YouTube videos, and they will be very helpful to you. So that's all about the syllabus. If you guys have any question, please never hesitate to contact me. You can send me email, you can call me, you can text me, and I will be very happy to help you. Okay, bye bye.